Hello everybody, my name is Robin Gray and I will be presenting over my topic about plant microbe interactions involving microbial volatiles. Plant microbe interactions may not be visible to the naked eye, but they occur in many different ways and on many different levels. Virtually all organs of the plant interact with microorganisms at a certain stage in their life, and this interaction is not necessarily negative for the plant. Indeed, there are plenty of interactions where the plants benefit either through direct or through indirect effects of associated microbes. Some microbes interact directly with the plant through a mutually beneficial manner, whereas others colonize the plant only for their own benefit. In addition, microbes can indirectly affect plants by drastically altering their environments. In these interactions, plants also serve as sheltered habitats for the microorganisms that may colonize apoplastic spaces, plant surface areas, and areas adjacent to the plant surface, such as the rhizosoil, the soil in the vicinity of the roots. And in a plant microbe relationship, there are secretions of different compounds known as volatiles that may be released. And the figure to the left shows bi- and multitrophic interactions that express volatile-based interactions with other organisms. The magnifying glasses focus on the presence of bacteria and fungi, which we will be discussing in a few minutes. Before we focus on relationships between plant and microbial interactions, I first want to get a good background on what volatiles are. Volatile organic compounds, or VOCs, are chemical substances produced and emitted by plants or organisms in a gaseous form. For example, in plants, when they react to stressful situations, they release VOCs for defense, targeting their predator, or priming themselves for protection. These volatiles are very useful as they are able to travel far from the point of production through the atmosphere, porous soils, and liquid. However, plants are not the only organisms that are able to produce or secrete these volatile organic compounds. Microbial organisms are capable of producing VOCs as well, and they are called microbial volatile organic compounds, or MVOCs. MVOCs are a variety of volatile compounds produced by fungus, molds, and bacteria during metabolism. There are more than 1,000 organic compounds that have been identified as MVOCs, which are comprised of alcohols, aldehydes, hydrocarbons, acids, ethers, esters, ketones, terpenoids, sulfur, and nitrogen compounds. It is difficult to make a reliable list of MVOCs for relevant microbial species, since different microorganisms could share the same MVOCs. Considering that only about 1,000 microbial species are currently described, which at least 1 million species are expected to exist on Earth, the VOC profiles of a surprisingly small number of microorganisms have been investigated so far, but they are still being slowly discovered every day. MVOCs being released may affect different microorganisms and cause many responses. The figure to the left are responses in bacteria, fungi, protists, and plants caused by microbial volatile organic compounds. The plant responses, such as development, growth promotion, and growth inhibition, are topics that I will be diving into as we begin to understand how microbial volatiles affect plant microbe relationships. Plant responses depend on how microbial volatile organic compounds signal. We want to understand that plant microbe interaction by analyzing through their microhabitat association through either bacteria or plant fungi. We know that MVOCs act as multitrophic signals communicating through their very complex systems, which is why we want to understand its microhabitat, as I said before, that bacteria and fungi relationship with the plant. By becoming aware of MVOC signaling, such as bacteria emission, for example, we can then pinpoint the active compounds responsible for VOC-mediated plant growth. Since remember, MVOCs for plant microbe interactions can result in possibly the alteration of plant root system development, plant physiology, and biomass production. Therefore, we will be looking into different studies that focuses on the relationship between microbial volatile compounds promoting plant growth and development. We will be discussing a study that focuses in understanding bacterial VOCs that promote plant growth involving indole mediating auxin signaling. Indole is an organic compound that is widely distributed in the natural environment and can be produced by a variety of bacteria. Auxin, on the other hand, is a plant hormone, which causes elongation of cells and shoots and is involved in regulating plant growth. The methods used to understand the effects of indole and auxin signaling. Bailey et al. Used to plant Arabidopsis thaliana lines, different bacterial strains, and isotopic labeling were used in observing their interactions. We will see the responses of A. thaliana in vitro in the presence of VOCs emitted by indole-producing Pseudomonas and Burkholderia species, along with the well-known indole producer E. coli. A little background on the methodology and experimental design. All experiments were performed with A. thaliana columbia ecotype line, which were obtained from Nottingham Arabidopsis Stock Centre in the UK. And the seedlings were surface sterilized and grown on parafilm sealed vertical plates. Meanwhile, bacterial strains, such as the indole-producing species, were cultured in Lauria bartania broth. While the seedling were sown in their culture, the Lauria bartania agar is placed in the middle of a stainless steel ring, which delimits the bacterial compartment. We can see this experimental design in the figure shown as Bailey et al. Provides a volatile source releasing VOCs, which will try to diffuse through a diffusion-proof compartment surrounded by a stainless steel ring. This ring separates bacterial strains, releasing the VOCs in order to only allow for gas exchange. Bailey et al. Is observing the bacterial volatile indole in triggering the increase of the lateral root number and biomass in Arabidopsis. Here we see the results of this interaction. Arabidopsis thaliana seeds were co-cultured for 21 days after germination with wild-type E. coli and its TNAA mutant. E. coli, remember, is an indole-producing bacteria. TNAA mutant is a devoid of tryptophanase activity and is unable to produce indole. In this part of the experiment, Bailey et al. Uses these bacterial strains and physically separates them from the plant by a stainless iron ring in order to allow only gas exchange. The other side of this figure deals with different concentrations of indole, which A. thaliana seedlings germinate on via growth medium supplement. After 14 days, the root numbers are calculated and the shoots were measured after 21 days for biomass determination. We see that E. coli was able to have a significant growth in both biomass, secondary roots, and root hair length, while TNAA mutant was not as successful, showing less growth in both biomass and secondary roots. Compared to indole growing with the plant instead of being separated, there is a tremendous growth in both 10 and 100 concentrations of indole for secondary roots. We do see an increase in biomass in the 10 indole concentration, but not in the 100 concentration. This may be because of too much high amounts of indole interacting with A. thaliana. This one example of how Bailey et al. Shows the interactions of indole affecting A. thaliana. We see how this affects the plant growth, which involves the plant hormone auxin. We saw that high concentrations of indole produces high amounts of secondary roots. A. thaliana was able to convert this bacterial indole, or microbial volatile organic compound, to auxin, and stimulate this plant growth hormone to increase its growth in both root and shoot systems. Not only does this example of MVOCs prove to affect plant growth, but there are also other examples of fungal VOCs interacting with auxin as well. In this study, we will see how microbial volatile compounds are produced by the diverse isolated species known as Fusarium oxysporum, which is a fungal species complex that often resides in rhizospheres of assorted plants. And we will observe on how strains of F. oxysporum VOCs will affect plant growth using A. thalania and tobacco. A little background on these isolates. They are soil-borne fungal species that causes diseases in more than 100 plant species by invading through the roots and subsequently blocking water and mineral movement through the xylem of the plant. However, for this study, we will see some similarities of how plant growth is affected by these MVOCs. As for the methods, Bittas et al. Used a variety of F. oxysporum strains, which were cultured on half-strength potato dextrose agar at room temperature. The seeds of A. thaliana ecotypes were acquired from Lay's Seed Co. from Round Rock, Texas. These seeds were surface sterilized, similar to the methods in which Bailey et al. Performed in their study. However, in this study, we see the culturing of F. oxysporum strains, and we'll see each plant remove its roots from its shoot. We also see them use Y plate-based Petri plates to help them determine whether CO2 produced by fungal culture contributed to plant growth, along with analyzing any VOC compounds by gas chromatography. While we observed in Bailey et al. Study the focus of A. thaliana interactions with indol-auxin interactions via the root system, for this study, we will turn our attention to the different variations of F. oxysporum interacting with the shoot system of tobacco. Here we have 4 Petri dishes showing tobacco shoots fresh weight. This was measured after 2 weeks of co-cultivation. The volatiles chosen were 2 growth-enhancing isolates that had exhibited the least variation, NRRL 26-37-9 and NRRL 38-33-5, and 1 neutral isolate NRRL 38-49-9, which were chosen for further analysis such as shoot fresh weight. We have our control with no fungus on the Petri dish and our 3 different strains of F. oxysporum. We see that as with the control, volatiles from NRRL 38-49-9 did not increase tobacco growth, but volatiles from NRRL 26-37-9 and NRRL 38-33-5 resulted in an increase of 2.5 and 3-fold shoot weights. So again, we see this relationship of how volatiles affect plant growth, which induces the plant hormone auxin. Overall, in this study, we are shown that volatiles from genetically and ecologically diverse strains are significantly enhanced by plant growth. This ability likely contributes to the rhizosphere competency by increasing nutrient availability. We also learned that because volatile metabolites can travel through air and porous soils, they can potentially function as signals for both short and long-range organismal interactions, even in the absence of water as a medium. We observed this through both studies on these 2 types of plants secreting MVOCs as they communicate at a distance through either their root or shoot systems. Finally, we have highlights and next steps. Not only does microbial volatile organic compounds have different interactions and responses, such as plant growth and development, which we have just discussed, but there are many other interesting factors that these MVOCs can associate with, such as interesting topics of how plant microbe interactions change plant VOCs emissions and reduce insect resistance, metabolizing carbon, chemoattractants, and signaling distances for communication are all different examples of how vast this research could be. Even though MVOCs are still being identified and studied, there is so much potential in understanding these volatiles which people are still looking into for agricultural applications. Since we continue to use old methods of chemicals to protect our crops, there is still a thought of replacing those traditional harmful synthetic fungicides with unique MVOCs to promote growth and protection. But overall, the study of microbial volatile organic compounds are still being studied and will probably be studied for a very long time. And finally, these are a few of my references, and I hope you've enjoyed my presentation. Thank you and have a nice day.